Hajar, today I am excited about this episode.
I could tell the way you came in.
I mean,
Excited.
we talk a lot about science on this show, right?
Yeah, we do.
And a lot of it is really amazing, like next level futuristic stuff.
Mind-blowing.
And I mean, look, even some of the things that have been around for a while, like, I don't know, MRI machines, modern surgical techniques. When you get into the actual nuts and bolts of how they work, it's amazing. These are some of the most incredible innovations, I think, in the entire history of science.
I agree. I love how excited you are about this. Where are we going with this?
Well, this is Smarter Than Cancer, by the way. I'm David Hoffman.
And I'm Hajar Aldas.
And in the last episode, you did a terrific job of introducing the concept of immunotherapy.
Why, thank you.
And this time, we are going to get smarter with a capital S, because I get to talk about one of those truly amazing scientific leaps, which happens to be the very latest thing in immunotherapy. It's something called CAR T-cell therapy, and I think it's incredibly exciting.
I do too. I can't wait to hear it.
So, to get into CAR T-cell therapy, we have to start by talking about T cells. These are a specific kind of white blood cell. We all learned in middle school biology class that white blood cells are the active agents of our immune system, the soldiers in our body's fight against disease, if you like. There are different kinds of white blood cells that battle different kinds of diseases. One of the functions of T cells is killing off cells with rogue reproduction errors, such as cancer cells. Sounds great. The problem is that the cancer cells that take root and grow tumors and go metastatic do those things because the T cells haven't successfully rooted them out. They make it so the T cells don't notice them, can't see them. They're stealthy. They don't have the characteristics that would make the T cells know to attack them. So what happens in CAR T-cell therapy, which is short for chimeric antigen receptor T-cell therapy, doctors take out some of your T cells, and then they modify them so that they will attack your cancer, and then re-inject them into your body. Here's Dr. Rainier Brengens, the chair of the Department of Medicine here at Roswell Park, and a leading researcher in the development of this treatment.
That immune cell would otherwise completely ignore the tumor. And so we are kind of re-educating those immune cells. So what we're talking about is, I guess, we're taking the cells out of the body, bringing them to school or to college and teaching them what in fact is a bad cell and what they should go after. So you genetically engineer them, which means that you can create in the laboratory a gene that encodes for a receptor, which when expressed by the immune cell, will now allow the immune cell through that receptor to recognize cancer cells.
Dr. Christopher Choi, Roswell Park's Senior Vice President of Industry Partnerships, is also intimately involved in developing and improving this treatment as the head of Roswell's GMP Engineering and Cell Manufacturing Facility, which is the largest academic facility of its kind in the country. Wow, so you're really training, if you extend the metaphor of the T cells being soldiers, this is like taking them out for special training and then putting them back in the field.
Yeah, I would say even more than special training. You're actually making these super soldiers. Absolutely.
So I'm picturing Captain America going into the thing and then coming out like twice as strong. And
Absolutely. So this is the beauty of a CAR T cell versus other types of immunotherapies like checkpoint inhibitors. These CAR T cells, that's where you have the antibody. You know the antibody recognizes the cancer.
Amazingly, they do this by actually modifying the DNA of these T cells.
We are modifying the DNA. That is absolutely correct.
How do you modify DNA in a living cell?
There's a variety of ways that investigators have used to introduce a gene into a cell. You can use electricity to get the gene into the cell. That's generally not a very efficient system. The irony actually is that investigators have co-opted viruses to do this.
This kind of sounds crazy, but they're actually using viruses to treat a disease instead of causing one. This works in this case because viruses are basically tiny little biological machines for modifying DNA. That's what they do. They're little parasites that burrow into your cells and change the genetic code to make the cell make new viruses. That's how they reproduce. So, long story short, these doctors were looking for a way to modify the DNA of T cells, and they said, why start from scratch if we don't have to? These viruses are really good at doing that. So instead of trying to imitate that ability, let's just capture it. Of course, before you can use a virus to safely modify a T cell, you first have to significantly modify the virus.
What you do is you actually take the machinery that these viruses use to introduce itself into human cells, and you co-opt that so that you can create a virus that is able to infect a cell, but the virus itself doesn't grow because all the machinery that the virus needs to proliferate or to cause damage, all that machinery has been taken out. Only capable of doing one thing, which is introducing the gene that you want into the cells that you're looking at. And so what you're left with is a vehicle, a viral vehicle that allows for us to introduce a gene or multiple genes into human cells, and in our case, human immune cells. You take a bunch of virus and you throw that virus onto the T cells. The virus finds its way to the T cell, inserts the gene, and then the virus is gone. You know, why reinvent the wheel? If the virus already finds an efficient way to introduce its own genes into the cell, we can use that same pathway to introduce whatever genes we want to into the cell. And the one type of virus that they use, called a lentivirus, is actually the same class of virus that causes AIDS, the HIV virus.
Wait. What?
I know, right?
I want to make sure I'm understanding this.
Yeah, absolutely.
So they take the same kind of viruses that cause AIDS?
More or less, yes.
And then they what? They de-virus them?
Like, they modify them, yeah.
So they're using HIV to cure cancer?
Kinda, yeah. Here's Dr. Bredgens again.
And that sounds scary initially, and certainly 25 years ago when we were doing our initial studies and we were proposing to do this, there was a, I guess, understandable but not scientifically reasonable argument that this might be dangerous.
And I'm seeing the sensationalist tabloid headline.
Oh, yeah.
Doctor says that AIDS is the cure for cancer.
That's exactly what happened. And so back in the early years when we were doing this and we were getting this technology, testing it, et cetera, that was the fantastic headline, is using AIDS to cure cancer. That's not entirely or even remotely true, but
sounds good.
Yes, it certainly got a lot more looks. If we had social media back then, you probably would have gotten a lot of hits. It would have gone viral.
No pun intended.
No, actually, no. That wasn't intended. Yeah, it would have literally gone viral.
As outrageous as all of this sounds, it actually works really well. This is one of the most promising new cancer treatments to come along in a long time, particularly when it comes to treating what are called liquid cancers, meaning cancers of the blood, things like leukemia, lymphoma, and myeloma. In the best cases that you've had here, you've read about, what's the result for the patient?
It's life-changing, and there are patients that we treated over 10 years ago that are still alive and well.
Dr. Marco Davila is Roswell Park's Senior Vice President and Associate Director of Translational Research.
So lymphomas that were not curable, that were incurable, are now curable with these gene-engineered cell therapies. And at the most recent American Society of Clinical Oncology meeting in Chicago, they presented 5-year data for myeloma for patients treated with CAR T-cell therapy, that after 5 years, they see patients still in remission. So now they're using the word, this is potentially cure for myeloma. So we've seen that for acute lymphoblastic leukemia. We've seen that for a non-Hodgkin's lymphoma, and now we're seeing it for multiple myeloma. So tremendous advances.
This is particularly exciting because until this treatment came along, the only treatment available to many of these patients was a stem cell transplant in the bone marrow, which is a really difficult procedure with a famously long and grueling recovery period. This new cell therapy is not a simple procedure, and it absolutely has challenges, some of which we'll talk about in a minute. But almost everyone would agree that it's much easier on the people getting it than a bone marrow transplant would be.
Patients, generally tolerate this therapy well. There's some CAR T cells that could be a little more toxic than others, and the readmission rates are higher. But in general, it's a very safe therapy, and the patients recover much quicker than with an autologous stem cell transplant or an allogeneic stem cell transplant.
I mean, years ago, when I was still training and I was starting to do this work in the laboratory, the head of bone marrow transplant was on a grant with us. And so I was kind of joking with him, telling him that my entire career goal is to put bone marrow transplant out of business. And he thought at the time it was funny. I don't think he's laughing quite as much right now. I mean, bone marrow transplant is still a very important part of what we do to treat blood cancers.
It's not an easy procedure to undergo, though.
No, by no means. And there are a lot of side effects with that as well. But, you could even consider bone marrow transplant to be kind of a very prehistoric version of what we're doing now with the CAR T cells, because that transplant also has T cells, and those T cells can sometimes recognize the cancer cells in the recipient. And that's the reason why they go into remission long term. So we kind of took out the middleman, and we just take a patient's own cells and make them recognize the cancer cells.
As exciting as all of this is, there are challenges. First, there are side effects that need to be considered. One of the most concerning is something called cytokine release syndrome. I think this is interesting because it points out a misunderstanding a lot of us have about getting sick. When we get a virus, like the flu for instance, the symptoms we feel, fever, chills, body ache, runny nose, all that kind of stuff, mostly aren't actually caused by the virus itself. They're mostly caused by our immune system. That's what a successful immune response feels like. But as we all know, some of those immune responses, especially high fevers, can be really dangerous in their own right. When a powerful immunotherapy like this one builds up a larger than normal immune response, larger than normal symptoms can come along with it.
Call that cytokine release syndrome. That is a fancier way of saying patients are having an immune response or an immune reaction against their tumor.
That means all your body, these T cells are attacking the cancer, and all these cytokines are being released, and it's causing this storm within this patient. And so the patient, look like they're getting sicker, but reality is it's your immune system that's working.
And it's highly variable. Some patients don't have many symptoms at all, and other patients can have very significant fevers. When we first saw this, when we injected our first few patients, it was both gratifying and obviously very alarming, because we had no idea if these fevers could get dangerous. And ultimately, you have to be very careful. You need to monitor these patients very closely. So if the patient starts to feel a little sick and starts developing fevers, you need actually quite specialized physicians to make sure that we can control those fevers.
Another problem here is that as well as this seems to be working for many of these blood cancers, it has not yet worked as well in what we call solid tumors, which is to say almost every other kind of cancer. Cancers of the pancreas, lungs, liver, and so forth.
So it works. And there are thousands upon thousands of people that are alive today because it does work. But it's not yet a panacea for cancer. It's not a cure-all. We're not quite there yet.
Here's Dr. Joyce Ohm, chair of Roswell Park's Department of Genetics and Genomics, to summarize these challenges.
So there's a couple big ones. So one, solid tumors are often really heterogeneous. So that just means that they're mixed. So they're not as straightforward as the blood cancers that all kind of look the same. When you get a solid tumor, the cells all look a little bit different. So that makes them harder to target. And they're very good at escaping from therapy. And so that means you have to really stay one step ahead of the tumors and find ways to target them in multiple ways at one time. So that's one of the challenges. Two, solid tumors have what we call a microenvironment. So they settle in your body and they're kind of shielded from the immune detection. So if you're using a cellular therapy that relies on the immune system to go in and attack it, and the tumor kind of forms this wall around it, that's not great if the immune cells can't get in and actually attack it. So that's really challenge number two, is you not only have to hit the cancer cell, but you have to break down kind of those very real physical walls around it and let the immune cells actually get in and attack it. And so one of the ways to do that is directly allowing the therapy to hit the tumor microenvironment, but also bolstering the normal immune response in the body around it. And so those are really important key pieces. And then we have to figure out how to stop the tumors from escaping from the therapy, which is one of the real challenges.
So there's a lot of things that are going on at the same time. You kind of have to learn a lot more, and we know a lot more now than we did when I started working on this, of the biology of a T cell. You need to understand more about the immune system and what the immune system does in solid tumors to protect the tumors from the killer cells. And, these are all concepts which, quite frankly, when we started working on this technology, nobody knew anything about. So over the 2+ decades that I've been working in this technology, what we know of the immunology of cancer, what we know of the origins of cancer, what we know of the immunology of immune cells has progressed tremendously. And so if the question is, do I think that this type of cell therapy will have application to patients with solid tumor? I absolutely do. And there are a multitude of laboratories now that are really focused on trying to understand the biology and trying to get this technology to work in solid tumors.
Hopefully, we're going to be enrolling our first patient for hepatoma within the next few months. And the ovarian cancer trial will hopefully be open by the end of this year, early next year. But we have other trials that will be opening hopefully by 2026 for breast cancer, sarcoma, and small cell lung cancer. And that's just the ones that are nearest in the pipeline to opening. We have a lot others that will be opening hopefully in a couple of years, and find those successes.
Another problem is that this is very new technology, and so the centers that offer it are few and far between. And it's also, at least for now, really expensive.
We make the cells for each individual patient, and you need to have a facility that is highly specialized, staffed by highly specialized workers to make these cells. That is not an insignificant obstacle.
So accessibility is a problem. The federal government and pharmaceutical companies right now have limited this therapy to only highly specialized centers such as Roswell Park. We're one of only 2 centers for the entire upstate New York area. So sometimes I have patients driving from 4 hours away. There's other kind of barriers. Need a caregiver because we're doing this outpatient. So we want to know if something's going on weird, in your house. We're not there to evaluate you. So there's some certain kind of stipulations.
And the cost of making these cells is very high.
It's expensive. Our cost, for patient care with the clinics, the hospitalization, stuff like that, add on to that. We've gotten better, at least at Roswell, as we've moved things outpatients. Outpatient costs are cheaper than inpatient costs. That is, I think, very helpful.
So that's another area that I'm really interested in researching is how do you actually make these therapies better and make the manufacturing more consistent? What are those factors that make a good T cell? Why does it work for some patients? Why doesn't it work for others? And what can we do in terms of that manufacturing process to make it more consistent and to make it more efficient?
But the more important thing is that people have done analyses because this is a curative therapy, potentially curative therapy. This could be a one and done therapy. So people look at the cost of going, okay, CAR T cells can be expensive. If you get this one therapy, let's add up all the costs of it compared to someone getting an infusion every 2 to 3 weeks for the next 10 years of their lives. Expensive drugs. Each infusion is expensive and that cost adds up. Patients getting hospitalized for, adverse effects, disease problems, things like that over years. And while CAR T is an expensive drug, it cures patients, or whether you look at a population of patients when there is a reasonable cure rate, the costs are actually lower than patients that are getting these infusions every few weeks. So that's a good cost benefit. But the thing that I didn't appreciate that I hear from patients is that they love not having to go to the clinic every week for blood checks and having the feeling miserable for 2 weeks after you get an infusion, then start feeling better that 3rd week just to repeat the process again. And that goes on for weeks, for months, for years. From a patient perspective, getting your life back is priceless.
So you said something in passing that was actually quite provocative. I think you said this is not yet a panacea for all cancers. That yet is a provocative statement. Do you think this has that sort of potential?
Well, yeah. I mean, I think that there are multiple objective observations that you could cite, work done in various laboratories around the world, that would suggest that the ceiling is extremely high, and that what's stopping us right now is not having a full read on how solid tumors evade immune detection and what solid tumors do to propagate themselves. But we know a lot more now than we knew 10 years ago or 20 years ago. And so what we're focused on and the type of next generation cells that we're making now address a lot of these limitations. And so that's very exciting. So I do think that there is reason for optimism. My goal is not just to get rid of bone marrow transplantation, but also I look for a day when we no longer give chemotherapy. Will we achieve that? I think so, but I could be proven wrong. There is a lot that we can do, and it could ultimately significantly change how we look at cancer and how we treat cancer. 20 plus years ago, when we were doing the first initial work on these CAR T cells, nobody would fund the research because nobody thought it would work. And nobody was really interested in looking at our results. It took a long time to convince folks because it seems like such an outlandish idea of how to use this type of immune-based therapy. And at that time, there may have been 10 labs in the world that worked on CAR T cells. Now there are hundreds of companies dedicated to this technology. The explosion of this technology is both gratifying and to a degree stupefying, because who would have thought that now everybody is looking at this technology to finally cure cancer?
Thanks for listening to Smarter Than Cancer, a production of Roswell Park Comprehensive Cancer Center.
The show is written and produced by your hosts, Hajar Eldas and David Hoffman.
Production oversight by Peter Sosha.
Additional logistics and support from Michelle Eisenstein.
Our production partner for this series is Citizen Race Car.
Post-production by Gregory Schweitzer for DCP Entertainment.
Publication and promotion supervised by Candice Chanteloup. If you or your loved one has been diagnosed with cancer and would like to schedule an appointment with Roswell Park, please call 1-800-ROSWELL. That's 1-800-ROSWELL. Or visit us at roswellpark.org.
If you're enjoying this series, we'd love to hear from you. Please reach out and tell us about your cancer story, and let us know what topics you'd like us to cover in future episodes by filling out the form at roswellpark.org/.
And if you know someone else who might find value in what we're doing, please tell them about it. You can listen and subscribe wherever you get your podcasts.
I'm just proud of myself when I understand something.
I think you've gotten a pretty good grip on this, actually.